Welcome, everybody. Thanks for joining us on this next session on day 2 of the 2021 Oligotherapeutics meeting. It's my pleasure to host this session. My name is Timothy Yu. I am a neurologist and researcher at Boston Children's Hospital and Harvard Medical School. And we've got a great lineup today. The theme of this session is rare disease as advocates. And in this, we have an absolutely stellar list of speakers who are going to be speaking about the application of technologies using antisense oligonucleotides to pioneer development of medicines for ever rarer conditions. Time is short and there's a lot to get to. So rather than drag this out, I'll dive in and introduce our very first speaker. This first speaker of this session needs no introduction. Dr. Stan Cook is known to all of you of Ionis some 30 years ago. Stan is one of the original pioneers of therapeutics and has led the development of over 20 markets in his career. He's received awards too numerous to mention, including today the OTS Lifetime Achievement Award, as well as more recently the Italian Best Biotechnology for Spinraza. In June 2021, Dr. Cook stepped down from his position as chair of the board of Ionis to focus solely on his scientific research and running Lorem, a remarkable new nonprofit that he'll be telling you about today. So without further ado, Stan, I'll turn it over.
Thank you for joining me. And thanks to the organizers for the opportunity to provide an update on the progress that we've made at Lorem. Lorem is a nonprofit foundation that I initiated in January of 2020. Our mission is to take advantage of the technology that we created at Ionis to discover, develop, and provide personalized ASO treatments for patients with ultra-rare mutations for free for life. And these patients represent, in my mind, the most underserved and desperately needy patient population that we have today, because they are unique. And in many cases, truly, there's a single patient with the mutation that they have that's been identified in the world. So they are entirely isolated. There is no patient advocacy group or other kind of support for them. Because of the significant delays in diagnosis and genetic characterization, which exist today, and in fact, the vast majority of these patients are never diagnosed. But if they are fortunate enough to eventually be diagnosed, that journey is typically several years and it's quite perilous. And so by the time we access these patients today, they're typically well advanced in their disease. They have an urgent set of needs. These mutations often affect multiple organs and provide diverse manifestations of the disease, most of which are severe. And because these patients are absolutely unique, there is no natural history that we can rely on. And yet, if we commit to one of these patients, we have to be prepared to provide our treatments for the life of that patient if the ASO works. So that's on the individual level. And then on the macro scale level, we now know that there are millions of such patients. So each of them may be unique, but the number of patients is enormous. And growing as we learn more about the genetics of these diseases. So those are the words. But I think it's a far better story told by the patient and parents of these patients themselves. And I think you'll agree. So I want to introduce you to the mother of one of our patients and one of our patients, Kelly Dalby, and her son, Connor.
Born at a few weeks old, we noticed a little bit off the way he was eating, When he was 8 months old, he started having seizures, and we started a different anti-epileptic, and nothing was really helping to reduce the seizure frequency.
Eventually, they learned the cause. The cause is unique. There's no other person in the world who has his mutation. Only through him hope for Connor.
And fighting for so many years alone with my son, and I wasn't very successful in helping him and Lorem with what is happening right now with a team of scientists and doctors and donors and making a medication child to help him and his quality of life and our family. It's still to believe and accept people are coming to help Connor.
Sadly, I have to tell you that Kelly and Connor's story is very common in this patient population, and there are stories that are far worse. And so I think we've made the case that this is a desperately needy, underserved patient population, and we're going to have to come together to find solutions for these patients. So I think the next step in addressing this issue is to define what it is that we have to have in place to actually try to do what we're doing. Well, first of all, we have to recognize that the vast majority of developed economies are in the same situation as the US, and that is that there is insufficient funding of healthcare to provide quality care to all patients with common diseases. And of course, these patients are unique. And so I think it's going to be very difficult to compete for those limited dollars if we use a traditional model. Second, you need a technology that can make it possible. Third, you need access to the patients and that are appropriately characterized and the physicians who can take care of them. And then finally, you need a supportive regulatory environment. Nothing is possible if the regulatory environment isn't supportive of this. And so to look at this circumstance just in a little more detail, there isn't sufficient money to go around, and certainly not sufficient for these patients. There is currently in the drug division at FDA no path to approval for these agents. And while we're very pleased that the FDA moved so expeditiously to provide guidance that provides a very clear path that we can take advantage of to treat these patients, that guidance is strictly for the charitable provision of these ASOs. And I'm quite confident that would change if a company tried to make a commercial opportunity out of one of these patients. And as I mentioned, these patients are spread around the world because most of these mutations are de novo, so there's very little clustering, and they come to us in very different stages of disease. So even if you could find a population of 30, I think the clinical trial itself would be logistically impossible. So clearly a charitable solution is necessary, at least today. Now, the second question is technology, and today the only technology for which guidance has issued for these patient populations is antisense, and there are good reasons for this. Of course, and the intrinsic efficiency of antisense technology was no surprise to me, but advances in the technology have supported the technology becoming much more versatile, and we have validated multiple routes of administration. We have a multiple mechanism that we could use, including mechanisms that actually increase the translation of specific proteins. And of course, we've validated that we know how to use these agents in a wide range of organs. I would add that the screening of ASOs to identify the optimal ASO for any individual target is complex. And to my knowledge, the only place where sufficient automation and experience exists to do this efficiently enough and rapidly enough to help these patients is at Ionis. In addition, we do know that within a particular chemical class, all of the agents behave very similarly. And we take tremendous advantage of that because that provides us information about what the right dose might be, what the dose frequency should be, what route of administration we use. And since we uniquely at Ionis have developed databases that integrate all safety observations in all controlled clinical trials and in our non-human primates, we can take advantage of that to actually know what adverse events that we need to watch for. So there's a great deal more than simply an efficient technology you need to have. Finally, we're focused on just a few organs, and those organs we know a lot about, and we know that the PSASOs distribute to them very well at low doses, and so we can take advantage of very low doses. I think that's important. These are the organs: central nervous system, liver, lung, kidney, and eye, by different routes of administration. And over on the right, you can see the total annual dose for a patient. And of course, they're very small, both because of the potency and the long duration of action. And so in a GMP facility, the smallest amount we can make really is about 10 grams. So given that these agents are quite stable, a single manufacturing run can supply a patient with the ASO that that patient needs for many years. And so that becomes not the super expensive part of the process that you might think it might be. Access to these patients and the necessary information to go to work to make a therapeutic is now available. Thanks to the pioneering work of the Undiagnosed Disease Network, and now the emergence of numerous personalized medicine centers in a wide range of academic centers. In fact, less than half of the applications we've received to date have come from UDN-affiliated organizations, and I think that's great news. It means that there is a great deal of attention being invested in academic medicine, in understanding and trying to find solutions for these patients. And of course, that's great. Without it, we couldn't do our work. I have mentioned that the FDA has been highly responsive, and I appreciate that tremendously. Definitive guidance on the preclinical requirements was issued on April of 2021, which is remarkably fast for new guidance. And that guidance was consistent with the proposals that we made and provides us the path that we need to make this feasible. Great news. So we do have the factors that are necessary to tackle this problem. And the simplest way to think of NLorem is that we are a bridge from the need, which is the patient and the investigator and the institution that can take care of the patient, and the engine for ASO discovery and development that we built at Ionis. So we provide a site to which the patient and research physician can apply for treatment. We have a process that assures that we make effective risk-benefit decisions. And then once we decide to treat a patient, we then provide funding to Ionis and the other institutions that we need to assure that the work is done. Of course, we direct the work. So with that, we began in January 2020. And I would say that despite COVID-19, we're off to really quite an extraordinary start. And that's great news for these patients. One thing that we were very fortunate to do was take advantage of two investigators, Tim Yu and Neil Snyder, and help them acquire ASOs and work with them during the process while we were setting up Lorem. And so even before we got underway, we had about 14 patients worth of experience under our belt, and that experience was tremendously important. We certainly thank both those investigators for allowing us to be a participant in that process. When I founded NLorem, I thought maybe in the first year we would have 4 or 5 applications because of the novelty of the situation and just getting the word out. And maybe over time that would expand. In fact, I expect by the end of our second year that we will have received more than 110 applications. I thought that perhaps we'd be able to treat about 10 to 15% of the patients. So far, we're able to treat about a third of the patients for which we are receiving applications. We continue to work with the FDA. We've made additional proposals that recommend some quality guidance for people who are making ASOs, and we also hope that we can have some relief on some of the CMC requirements. We've worked very hard to expand stakeholder awareness across all the domains that exist, but we've just really only scratched the surface. And you'll hear that term from me about 10 times before we get through here. We have just scratched the surface. Importantly, we've worked very hard to construct quality systems. Why is that important? Because we have very minimal preclinical data that we develop before we start treating these patients. And so that puts an even greater premium on quality at every step, a quality risk-benefit decision, the highest quality ASO that has been optimized as well as we can do it at Ionis. And then a specific treatment plan for each patient that outlines the clinical measures that we'll use, that assures that we're treating the patient for the right reason and the right way, and that we're going to be able to evaluate it, both as a natural history while we're preparing the ASO, and then in the 1-year clinical trial that we run post initiating the treatment. And of course, after that 1 year, if the drug appears to be benefiting the patient, then we will continue to follow it up on an annual basis. Because of the extraordinary demand, we've had to move much more rapidly in building our organization, and I'm pleased to say there's been an enthusiastic response from everyone, including the people who've joined us. And as you might expect, as more and more academic institutions are involved, centers of excellence are emerging sort of naturally. And of course, that will add to the efficiency of the process. We have achieved very broad positive coverage in the scientific and trade press, and we're still working on broadening awareness in the lay press. We had not planned to raise additional funds in 2020 because I wanted to make sure that the system was really working, but we became convinced that it was very early on, and so we've been fortunate enough to enjoy many new donors and many new contributors and many collaborators already who've joined us. And one of the more interesting features of this is that we have multiple sources of collaborators and funding. Biotech companies are natural partners, as are pharmaceutical companies, especially those in rare disease. Numerous investors in biotech have joined our effort. We've only begun to focus on charitable institutions, but the progress here has been very gratifying. And essentially all the vendors and CROs and so on that we depend on have stepped up and contributed either cash or cash plus in-kind contributions or in-kind contributions. And here's a list of some of the collaborators that we have already. And that, again, is great news for these patients. And Lorem cannot do this alone. Our goal is to create a network of collaborations broadly across all of the stakeholders, including the patients and parents, in this endeavor. Only that way can we manage to treat these patients with the kinds of efficiency and care that we need to exercise. I find it remarkable that during COVID-19, the total value of the contributions that we've received is well over $30 million already. And that's, again, great news. But the demand says that we're going to need to raise another $30 to $50 million in the next 3 to 5 years or to do what we need to do. But I would say that the progress that we've made gives me a great deal of encouragement that we can raise the money we need, and so that we will never have to tell a patient we can't treat them because we don't have the financial resources. So I do believe that Lorem is a sustainable solution. We have succeeded, I think, in having systems now in place that should maximize learnings from each patient. We owe these patients that, and they're just extraordinary opportunities to learn all sorts of things here. And again, we take advantage of the time that it takes to create the ASO, to collect a detailed natural history in that particular patient that then we can compare to the treatment period. Of course, we're going to encourage our investigators to publish all the case reports, and then we will publish aggregate results every year. We'll share those with investigators, with patients and parents, and all of the other community, so that we can all benefit from what we're learning. I want to just spend finally just a couple of minutes on the roles of Lorem. Of course, we have some short-term roles to play than we're playing, but I think where the leverage will come is in the long-term roles that we're going to play. So our first goal is to treat the patients that we can help today. That is an axiomatic comment for therapeutics. Treat those you can help today. And we're doing it for free for life. And we believe, as Lorem matures, that we can translate that into thousands and thousands of patients we treat, but not millions. And that's an important point. We think quality is essential, and we have worked very hard to be a pioneer in quality systems that we hope others will follow. And I also strongly urge everyone involved to recognize that screening ASOs is a process, and to identify the optimal site and the target RNA is a complicated process that to date is done best at Ionis. We're committed to maximizing learnings, and so all of those are roles that we can play immediately. And in addition to that, establishing a broader and broader network of collaborations that then takes advantage of all the various people who are interested in this idea. So those are short and near term, but in the longer term, where the real leverage comes, is in the creating of a roadmap for others to follow, and a quality model for others to follow as other technologies matures, gene therapy, other approaches, and so they can come online. Of course, we'll work hand in glove with those other foundations that may get set up in the coming years. And I think in that way, then we can look forward to a day when what we've done with Lorem can be leveraged to move from thousands of patients to maybe millions of patients treated worldwide. And that'll be a great day. We also will be driving, I think, solutions that are more holistic and more healthcare wide, that would include the incorporation of genetic screening in newborn screening efforts. I mentioned that we have established this network already, even though we've only scratched the surface in all of these areas. But the impact of those collaborations, in addition to cash donations, has been a 40% reduction in the cost per patient. And every dollar that we can save for patient A is a dollar we can use for patient B. So we think Lorem is the right thing to do. We think that we are pioneering a novel, non-commercial solution to a patient population that is not possible to address commercially. We think that we can pioneer for the entire space and lead to much more holistic solutions for these patients over time by serving as a roadmap maker and a model. And we look forward to continuing to tell you about our progress. And certainly we seek your support and your help in every way possible. Thank you.
Fantastic. Thank you so much, Stan, for this incredible effort that you've marshaled yourself and with the volunteer efforts of so many folks at Ionis. It's been a pleasure to see this effort grow. Let's few questions.
I'll be delighted. And by the way, Tim, thanks for asking me to speak.
Our pleasure, our pleasure. So a couple of questions from the audience. So let me start with by combining two questions from Dan O'Reilly and Alex Garanto. Dan O'Reilly and Adam Klaus. Explain how you select patients. The number of applications that you cited is tremendous. How are those sifted through to identify cases that are appropriate? And one perhaps follow-up from David Corey, in particular, if a clinician has investigated the genetics through sequencing, but doesn't have the basic science expertise to understand the mechanism, how might Lorem help?
Okay, so the first question really is a fairly complicated process. As I've stated in every presentation I've made, we have significant limitations with regard to technology. And we are only focused on 5 organs and 3 roots. Those are the organs we know, the organs we get great potency in, and that allows us to assure that these patients are exposed to only the most prudent risk. So the technology is not capable of making a new gene or de novo creating a protein. So any patient with a null mutation is not a candidate for our technology. Of course, I've been involved with as many gene therapy organizations as possible to see when we can bring that sort of thing online. In addition, we need to look at the type of mutation we have, ask what kind of mutation it is a gain of function, loss of function, splicing, a variety of questions. And then we hone in on whether we believe that we have a design ASO, a type of ASO benefit to that patient. And so the 40% acceptance rate is quite a little bit higher than I had expected to begin with, as is the demand. And once we, as technology experts, work through that, then the case is presented to the committee I created called the Access to Treatment Committee, which is made up of all the expertise that we think we need to manage these decisions. These are complex risk-benefit decisions. And so it's primarily driven by the technology. An additional consideration that has happened quite a number of times is that the population of the mutation is significantly larger than we can handle. And we are limited to that size because we think any population larger than 30 in prevalence around the world can probably be better handled commercially because we are pristinely nonprofit. So, I mean, first question. The second question is that we require a great deal of information, and not just the sequence, but we need to know the nature of the mutation. We need to also have an institution capable of taking care of that patient and an investigator who can take care of that patient and perform the clinical evaluations that are important. So my would be to direct the patient to one of the sites that is set up to do this. There are many personalized medicine sites in the country and more coming online every day. And in fact, I think, as I said, we've gotten a larger number of applications from institutions that are not a part of the UDN, which is a bit surprising to me, but that's what you have to do. One of the problems that we've is that we get a lot of applications and people who are asking questions like what you asked, David. And so we've been trying to facilitate their finding one of these centers that's capable of helping them over time. And I hope not too long, I mean, sometime in the next few months, develop a roster of sites that are coming online and focus on helping these patients. And so at some point here, sure on our website we'll provide some solutions for people that would be in their local area. That's taking time because it's a complicated equation and there are a great many resources needed. So refer the patient. The art form here, by the patient and refer that patient is capable of taking care of it.
Understand. I think in the interest of time, there are a number of other questions flooding in, but I think we'll have to keep moving so we can, we'll make sure to cover those topics in the panel discussion, absolutely, so we won't lose them. So in order, let's move on to the next talk. My pleasure for this session introduce to you Bob Brown. Bob Brown, Dr. Brown is the Leo and Therese Chair in Medical Department of Neurology, Massachusetts Medical School. Bob is a physician, scientist, and neurologist who's leading the one. He discovered SOD1, the very first gene linked to the inherited form of ALS. Has been extraordinary honor being inducted into medicine. A pleasure to introduce and someone, Dr. Brown.
Thank you. It really is a pleasure to be part of the session. I want to thank you and the organizers for this opportunity, and also for the chance to hear these extraordinary other panels today about our own experience in generating antisense oligonucleotide ALS, focusing on two genes which have been at the screen, C9orf72. My work reflects the individuals. So then the remarkable who have these ASO studies, Orova, Jonathan, collaborators. Just say briefly that, of course, ALS disease characterized by motor neurons in the brain record relentlessly over 3 to 4 to 5 inevitably. In the early we with Tipu Siddiq and many the view that if we could 1% of cases that we might understand all ALS, both familial with the thought of ALS gene and their extraordinarily enough, probably 60 or 70 confirmed ALS genes. Most of these are dominantly transmitted. And indeed, it turns out to be the case that some 43 now have defined convergent points and sporadic ALS, among other things, through 2 and Stathman 2. So we are indeed beginning to see ways in which genetics to treating the entirety of ALS. But what's clear is that because these are transmitted for the most part, modulation have a role for those. The key point to make, ALS itself is when one takes genes that reflect 1 or 2% of the familial ALS genes, there are specific mutations within each of those. It's clear that most familial ALS represent extremely ultra-rare opportunistic intervention. Now, the most common gene that has been identified so far is 72, and I will speak about work that at least shows how one can go forward in terms of generating therapeutics, focusing on suppressing. For those who may not know the story, the C9orf, a gene on chromosome 9, which encodes a protein that's probably important, among other things, in vesicular and lysosomal function. It is the case that in the familial ALS cases with C9orf72 mutations, there are large expansions of a G4C2 repeat in exon, just after exon 1 and intron 1, which can be identified by PCR methods or by southern blotting. Some of these are enormous, in fact, representing hundreds or even thousands of expansions, whereas the normal would be perhaps 25 or 30 at most. And what's been very exciting to learn about this is it is the case. Expanded tract makes an RNA expanded in which has a pathway exit the nucleus, engage the ribosome, and make small peptide proteins, 5 of them, in fact, from each possible reading frame and the antisense directions. And these, in fact, may key toxic elements in this disease. They also are important in function and expression of the gene. The gene generates 3 RNA transcripts. 2 of them, V1 and V3, in fact, harbor the expansions. V2 makes the most abundant transcript and produces about 90% of the protein. So the goal then in thinking about how to treat this disease in a mutation-specific manner is to try to silence or suppress V1 and V3. And we undertook this with the brilliance of Jonathan Watts, Helene Tran, Michael Mozami, and others. And again, the goal then is to be left predominantly with V2 transcript expressed from the C9 locus. And I want to point out that many other people have been working on this. Ionis is well along in a program with Biogen testing their ASO suppressing C9 in clinical trials. With Jonathan Watts, we generated multiple ASO gapmers, as shown here, with varying chemistries, and I'll put these through a variety of tests for feasibility of suppression of C9. So, for example, shown at the top is an experiment with patient-derived fibroblasts, in which it was clear that 2 chemistries, the locked nucleic acid, and as shown here, the OMOE or 2'-OME chemistries, both could effectively suppress the offending transcripts in fibroblasts, and that was also the case in neurons generated from our mouse model of C9orf72. As Jonathan worked through these chemistries, it turned out to be quite intriguing that the position of the and the chemistries of the internucleotide bonds were quite critical, both as determinants of maximal efficacy and also toxicity. So without belaboring, I would draw your attention to what we're calling ASO52, which you can see illustrated here on the right, in which there are 2 critical substitutions of phosphodiesters for phosphorothioate bonds, which are quite critical in maximizing efficacy, as seen in our mouse model, looking both at cortical and spinal cord samples for suppression of the offending transcripts. So in fact, this ASO52 was taken forward as our lead ASO. When studied for dose dependence, it showed clear evidence of selectivity for V1 and V3, as shown in panel A, with an ED50 of about 4.8 nanomoles. It also was the case that there was a remarkable ability to suppress the R marker for activity of the gene, the poly GP dipeptide, as shown in the upper right. Time dependence was also studied in some detail, and it turns out that 30 nanomoles infused into 1 ventricle shows suppression of the offending transcripts and the poly GP dipeptides out to 20 weeks. We then showed that this was non-toxic both in sheep and in monkeys, and then have now used this to treat 1 patient, a 60-year-old individual with a very positive, unfortunately, sadly positive family history of C9orf72 mediated familial ALS. And I'll cut to the chase here. What this depicts in the x-axis is the time of treatment of this individual. And what you see in the y-axis in red here are the levels of the poly-GP dipeptide. And the point to make is that as we escalated the dose of this particular ASO52, we have observed about 80% suppression of the GP levels in the spinal fluid. And thanks to Fenbiao Gao, Kenkel, and others at Biogen, we now know that, in fact, poly-GR and poly-GA also fall off perhaps more rapidly than poly-GP. I won't go through too many additional details, except to say that at 2 milligrams per kilogram, we had a steady state level in the spinal fluid of the ASO. And it turns out that when we model this with both a rapid clearance and a slow clearance phase, we could quite nicely reproduce through prediction the actual values that we observed. Let me turn briefly to another project going in which we've tried to show ASOs very rapidly to move from through preclinical to clinical application. Briefly, STAthmin-2 is a protein that stabilizes microtubules, expressed prominently in motor neurons, and is very important in maintaining the integrity of motor neurons in the neuromuscular junction. Its status is determined by TDP-43 to control splicing so that a full-length STAthmin-2 is normally produced in the locus. When TDP-43 is disturbed, which is true in most cases of spread, many familial cases, as well as C9 ALS, there is missplicing of STAthmin-2. A cryptic form is generated, which leads to, at the very least, STAthmin-2. This was found through incredible work by Kevin Egan, Don Cleveland, and their associates, and Klim. Looks at the cryptic form of STAthmin-2, one can see it in many cases sporadic ALS as well as C9 or 72, although not in SOD1 ALS. So the question is, could an ASO be developed which is a surrogate for TDP-43, such that we could use the ASO, much as one sees a TDP-43 naturally performing the function, to lead to normal splicing of exon 1 and 2 and absence of the misspliced fragment. So just briefly, the cryptic region of STAthmin-2 has many potential sites for intervention. Jonathan Watts and colleagues generated more than 50 ASOs to attempt to find those that might correct the missplicing of STAthmin-2. We were privileged to work very closely with Joe Klim, and Kevin Egan himself paid attention to this work to study a model in which, with TDP-43 in human motor neurons, it was to basically TDP-43 suppression of STAthmin at the same time to generate high levels of STAthmin-2. And so this became a model for screening those. What is a normalized log scale of cryptic STAthmin-2 as responding to both non-targeting ASOs and ASOs generated in the Watts laboratory. And briefly, there's a dose-dependent suppression of the cryptic STAthmin-2 with 3 of the ASOs of particular interest to us. I can tell you that the same ASOs suppress the cryptic, increase the full-length STAthmin-2 and increase the levels of the protein. ASO1 here, for example, essentially generates or activates STAthmin-2 to full levels as compared to when it is in a motor neuron that's been silenced with TDP-43. Time does not allow a full discussion of the fact that the same ASO also rescues a deficit in axonal regrowth after injury. So with this as a background, we took the best ASOs and did a non-GLP rat study. There were 3 or 4 of these, and then selected the number 1 lead and did a 90-day rat study, which suggested that this was non-toxic and could be put into a human trial. And with FDA approval, we've now initiated a single patient compassionate use dose escalation study of this compound. Up there, let me just clear that analysis of gene defects less both helped and has way for the generation of personal biological intervention. And as we've tried to STAthmin-2, I think it's possible essentially clinical work very rapidly setting and with work that's being done by or in individual laboratories remains to be seen, but forms will allow us to rapidly generate therapies for these diseases. I'll stop there. Thank you.
Fantastic, Bob. That's just amazing, and amazing to see how quickly scientific input is being translated. So fantastic work. Why don't we take some time to take or two? So I'm looking here in the chat. Here's a question. This is from Athaya Almano. Good. Wonderful talk. What is the half-life of the drug ASO52 after administration?
Yeah, so the half-life upwards of 7 or 8 days. I can tell you, as I tried to suggest, there are two phases. One is most of it is very rapidly cleared. I mean, literally within minutes to hours, but then there is a slower phase, which allows the infrequent dosing.
Excellent. Thank you, Bob. And then another question from David Corey. What patient outcomes are you measuring in this trial?
Yeah, so this is a crucial point. So let me be very, very clear. What we have measured in that N=1 trial is only one major outcome variable, and that is the CSF level of the poly-GP dipeptide. Of course, many other aspects, including clinical parameters, but what I would argue strongly is that with an N=1 trial, we really can't draw any conclusions yet on clinical responsiveness, but I'm convinced that the parameter does show efficacy.
Fantastic. Thanks so much. Thank you for the great questions. And I'll move on and come back to you again in the panel session.
Thank you.
So moving on to the session, my pleasure to introduce to you Dr. Liz Berry-Kravis. Liz is professor of pediatrics, neurology, and biochemistry at Rush University Medical Center. And there she is of the Comprehensive Fragile X Clinic and Research Program at Rush. It's my pleasure to have been a colleague of Liz's. I've been so impressed. She and her research group have been at the absolute forefront of basic science and clinical translational work, not only into Fragile X, but neurodevelopmental disorders as well. And reflective of that, Liz is going to be speaking to us preliminary studies on use of ASOs to reverse imprinting and treat Angelman syndrome. Thank you, Liz.
I'm pleased to be able to share our early experience with an investigational ASO for disease-directed treatment of Angelman syndrome. This is an ASO called GTX-102. This is a presentation of the first 5 patients treated with an ASO to reactivate the paternal UBE3A gene in Angelman syndrome, and the work was all done at Rush University Medical Center in partnership with Genetics and Ultragenics. So Angelman syndrome is a severe form of intellectual disability with expressive language. Most patients have very delayed motor skills, ataxia, and tremor. They can have a happy affect and often have laughing spells, seizures, sleep problems, behavior problems that include separation anxiety, hyperexcitable behaviors and aggression, as well as excessive sociability, and have dysmorphic features such as a broad mouth, which are highly variable from patient to patient, as you can see here. And Angelman syndrome results from a defect of the maternal UBE3A gene. And the UBE3A gene is imprinted, but not in other tissues, such that the paternally derived UBE3A is shut down, and normally only the maternal UBE3A gene is expressed in the brain. UBE3A cause Angelman syndrome, and these can be a maternal deletion in the 15q11 to 13 region, paternal disomy, in other words, 2 chromosome 15s from the father, such there's no maternal copy of UBE3A, and an imprinting center mutation that causes the maternal gene to be shut down and behave like the paternal gene, as well as mutations in the maternally derived copy of UBE3A, rendering it non-functional. Patients with uniparental disomy and imprinting center mutations and some UBE3A mutations tend to be a little more high functioning than deletion patients. However, all patients are fairly severely affected, and we through methylation PCR to show a pattern of only the paternal allele present, FISH or microarrays to show the deletion, UBE3A UPD studies, and imprinting center sequencing to figure out exactly what type of mutation a patient has. Now, in the normal situation, we understand that the reason the paternal UBA3A gene is shut down is because of active transcription of an antisense transcript, a UBE3A antisense transcript that interferes with transcription of the gene. While on the maternal chromosome, the antisense transcript is shut down, and so the UBE3A gene can be transcribed. In the situation, when the maternal chromosome contains a mutated or absent UBE3A gene, then no UBE3A can be made because the paternal gene is shut down. However, we can apply an antisense oligonucleotide that prevents transcription of the antisense gene and then allows transcription off the paternal gene, and theoretically that would be a genetic correction for Angelman syndrome. Oops. So this has in fact been tried in the Angelman in Meng et al. In a paper in Nature in 2015, and here you can see that the UBE3A gene RNA is increased and the antisense RNA is decreased in the presence of the ASO. The UBE3A protein can be seen in the presence of the ASO and is expressed in brain. So this appeared to be an effective strategy for UBE3A expression in the mouse with Angelman syndrome, and therefore is now moving into clinical trials in humans. And so GTX-102 was the first ASO that has been tried in Angelman syndrome, and it targeted the cluster 2 region of the antisense transcript because this was a conserved area and likely the origin of the transcript. It's at the beginning of the transcript. It's structurally different from cluster 1, has highly evolutionarily constrained exons, suggesting a regulatory function, and exons are identical in humans and non-human primates, allowing pharmacodynamic studies in a non-human primate model. And targeting the exons looked like that would increase the chance of terminating transcription, since the introns would be spliced out during transcription. So this GTX-102 was first used in humans starting in about February of 2020. And this is a first in human study associated with 4 monthly doses with a dose escalation within cohorts across multiple cohorts. There was no placebo control group, and the ASO was given intrathecally by lumbar puncture, and it consisted of a screening, treatment, and follow-up period. Was the safety of GTX-102. Secondary was the pharmacokinetics in blood and cerebrospinal fluid, and exploratory outcomes were measures in different functional domains of Angelman syndrome. Patients were included in the study if they were between 4 and 17 years of age, if they had a deletion, stable seizure control, and generally healthy and able to tolerate anesthesia. And they were excluded if they had had recent changes in medications or diet, or if they were not able to ambulate independently, even with an assistive device. The patients enrolled in the study were variable in terms of their severity. These are ratings on the Clinician Global Impression Severity Scale, which has been anchored to Angelman syndrome, such that a person mildly affected would mean mild for Angelman syndrome, not mild for normal. And so these patients varied in their severity in the different domains. There was a dose level that increased in terms of the starting dose in each cohort. So for instance, a cohort would come in dosed at 3.3 milligrams, and then there would be a DSMB review, and then they would get a dose of 10 milligrams, and then there would be a DSMB review, and then they would get a dose of 20 milligrams. And then the next cohort would come in starting at 10 milligrams, for instance, and then escalate to 20 and 36. We actually only enrolled 5 patients, 2 in cohort 1, 2 in cohort 2, and 1 in cohort 3 before the study had to be stopped for a serious adverse event. The patients that were enrolled, all 5 were treated at Rush. Opened prior to the COVID pandemic. And so we enrolled all of the first 5 patients. There was an 11-fold difference between the lowest and the highest dose used in treatment. And these patients got a variable dose amount before the study was stopped. So the pharmacokinetics from these 5 patients appeared to be dose proportionate. In other words, the patients that received the 3.3 dose had the lowest increase in the plasma GTX-102 concentration, and then the 10 milligram dose was more, and then the 20 milligram dose was higher. There were some adverse events that we saw transiently after the LP infusions of the drug. These were dose related and primarily observed at 20 milligrams and higher. Variable severity between patients, and there's this kind of an ataxia and fatigue symptom that started 2 to 6 hours after the injection, lasted about 1 to 3 days, and patients were back to normal after it resolved and were able to eat and interact normally. They were just somewhat wobbly, off balance, and a little tired. There were other mild events like headaches, UTI, and typical childhood infections, and the patients tolerated LPs and anesthesia well. We then ran into a serious adverse event of lower extremity weakness, which was mild to moderate in severity. It was seen in all 5 patients, occurred 6 to 30 days after the last infusion. Resulted in significant weakness in 3 and complete inability to walk in the 2 youngest patients. In 4 patients, it occurred after the highest dose, and in the smallest patient, it occurred after the second highest or 20 milligram dose. Dosing was paused for all patients after the first case was observed, and the most severe aspects lasted 1 to 3 weeks, although full resolution took up to 86 days. However, clinical improvements that we saw in the patients were sustained longer than the serious adverse event. So this is just what like on MRI. This was inflammation of the nerve roots that you can see along here. And then these are the individual nerve roots coming out that are enhancing, consistent with inflammation. This was associated with a CSF protein increase, and we viewed it as an acute radiculopathy. It did not appear to be clinically similar to the post-dose ataxia, and the severity of the post-dose ataxia in a given patient was not predictive of the severity of the delayed nerve root weakness. So we have that we're looking at areas like sleep, seizures, communication, behavior, cognition, and motor functioning. And in fact, these patients showed a great deal of improvement. This is the CGI-I, which is the clinical global impression that takes into account all of the things that the patient is doing or that are reported to the clinician from the family. And so a rating of 3 means this is a really knock your socks off kind of improvement, and meaningful improvement in a rating of 2, meaningful improvement in at least 2 areas, and a rating of 3, a meaningful improvement in 1 area, or more minor improvements in say 2 areas. So these patients all improved in multiple domains a great deal, and in fact these worsening scores were when the patients had the acute inflammatory polyradiculopathy, and so they were worse in gross motor because they were not walking as well. So what we saw was things like acquisition of spoken words, signs, and gestures, increased use of ability to respond to the name, follow commands, and focus on tasks, ability to feed the patient, could feed themselves with a fork when they couldn't previously do it, point to AAC items, ability to catch a ball. One patient that learned to swim on her own in about a 2 to 3 week period. Improved gait and posture, ability to go up hills and walk over uneven surfaces, walk on sand, stopping and turning better. So lots of improvements. Improved sleep for the patient with the worst sleep at baseline, and improvements in maladaptive behaviors and increased social engagement and playing games and imitating. This in the Bailey, we did in domains in several patients on the Bailey, and the Bailey also picked up the gross motor worsening when the patients had the acute inflammatory polyradiculopathy. And these improvements are way more development than you would ever see in Angelman syndrome, because this is, for instance, a plot of expressive language in Angelman syndrome from age 2 to 16, and you can see it's very flat. There's practically no natural development. The ORCA is a communication measure developed for Angelman syndrome to identify improvements in language in patients with very low levels of language skills. And so this measure did improve in 3 patients, a significant improvement of over half a standard deviation, and was stable in the other 2 patients. We did EEGs, which showed an improvement in notch delta waves, which are a common finding on the Angelman syndrome EEG, and improvements in epileptic discharges in 3 out of 4 of the patients. We also saw an improvement in delta power in 3 out of 4 of the patients, and delta power has been correlated with level of function in Angelman syndrome. The Actimyo is a device that patients wear around their ankle, and it measures the amount of walking they're doing. And typically Angelman patients are somewhat below normal controls in terms of distance walked, but we did see improvement across the study as denoted by these points going up as the Angelman patients were walking more and were more stable. So it looks like this measure showed an improvement as well. Also, clinical improvement with a relative mean CGI increase of about 2 point. All patients had at least that were much improved or better. The very much improved scores. There were improvements on the Bailey, the ORCA, EEG, and potentially the improvements began at the lowest 2 dose maybe. We had necessary doses, which are the most in the youngest and smallest patients, of to be local to the nerve root, where the Dr. Mayo and these patients outlasted the adverse. So the protocols be amended to use a lower dose of 3 milligrams, depending on the age of the individual. Individual dose titration and will be capped at 14 milligrams. Administration procedures being modified to try to eliminate and reduce the duration of exposure at the injection site in Dellenberg. And this new protocol will be done in Canada because the FDA has, and we're still working with FDA before dosing and enrollment in the USA. And so there's more to come, and hopefully we'll have the opportunity to see how this drug works in long-term treatment. And it seems that this study has actually provided early evidence that antisense oligonucleotide to decrease the antisense transcript and reactivate the paternal UBE3A allele may be effective in ameliorating symptoms of Angelman syndrome. Thank you.
Fantastic. What a really stunning set of results. Thank you so much. As a neurologist who's got a great deal invested in the care of patients with neurodevelopmental conditions, I just want to comment and say that the gains that you've shown are possible with intervention and really answer a longstanding question about what the treatment window is, whether it's still open in many of children with conditions like this. I just wanted to applaud you on that. So let me just pause and take some questions here. First is, do you have a hypothesis the cause of the more severe adverse event seen in these patients?
Well, we think it's a direct toxicity. We sent extensive batteries, antibody testing and tests to look for autoimmune phenomena, and those were, we could not find any kind of antibodies positive anywhere. The CSF protein went up, and we could see the enhancement around the roots, and we think that it was just the drug is concentrated, it sits there when you inject it, and it was probably a direct toxicity to the exact mechanism of that. We don't know. But doses equivalent to this, where doses actually 10 to even more times higher than this, were used in the monkey studies. Any abnormality. So that because Angelman patients are undermyelinated, it may be easier to have toxicity from an ASO for them.
Those are really important considerations. There's a follow-up question. Was there any evidence of innate immune?
No, we really did not see any evidence of and we sent a variety of different bands as well as ganglioside antibodies, as well as blood markers like sedimentation rates, and nothing, it was really very clean. Nothing came back as abnormal.
Very important to investigatory work to follow that up. Thank you. There's another question here about the some details of the ASO and the type of chemistry used and the length, perhaps.
Be things that so it's a purple, and so I haven't been told everything about it. I know the location that it targets, but other details are not known to me.
Understood. And is perhaps those who may be contemplating use of ASOs for other indications like this. In this case, were the ASOs take vivo proof of concept cell directly into patients, or was there the intermediate of proof of concept in other?
The ASOs were studied in the mouse with Angelman syndrome, and then there was extensive toxicity testing in non-human primates before they went into humans. And this ASO does it reduces the UBE3A antisense and increases the UBE3A protein in those models.
Thank you, Liz, for highlighting that. That's important proof of concept. Impacts the risk benefit as you're contemplating. All right. Continuing on, I'd like to and again, we'll save questions more for the panel and move on to the next of the session. And that will be by Pushkal, who is the Chief Medical Officer for Alnylam. Alnylam needs little introduction, but we all know them as a leader in the field of RNA interference. And Dr. Garg himself has over 20 years of clinical drug development with prior stints at Bristol Millennium. And he's going to be telling us about an RNAi approach for the treatment of primary hyperoxaluria.
Everybody, my name is Pushkal Garg, and I'm the Chief Medical Officer at Alnylam Pharmaceuticals. It's a real pleasure to join you today for this meeting. And I'm grateful to the meeting organizers, and especially to Tim Yu, for inviting me to this session on rare diseases and the role of patient advocates. Today, I'd like to present a case study on lumasiran, a medicine we use for primary hyperoxaluria 1, as well as other indications. It really highlights the critical role that the advocacy community can play in helping to get a medicine approved for patients with a serious rare disease. As I'm sure most in this audience know, Alnylam is pioneering a new class of medicines based on RNA interference, which we applied to developing a treatment for this devastating disorder, primary hyperoxaluria type 1, or PH1. Now, PH1 is a rare autosomal recessive disorder of oxalate synthesis. The elevated levels of oxalate lead to the formation of kidney stones, calcification of the kidney, which is known as nephrocalcinosis, and ultimately kidney failure. Once the kidneys fail, oxalate accumulates in the bone, heart, eyes, and other tissues, causing a serious condition known as systemic oxalosis. Unfortunately, there are very limited treatment options for these patients, with the only curative option being dual liver kidney transplant. Hence, the unmet need here is enormous. The pathophysiology of this disease is shown in a bit more detail in this cartoon. The mutation that causes PH1 is in a liver-expressed gene called alanine glyoxylate aminotransferase, which encodes for a protein called AGT. AGT normally converts glyoxylate to glycine. But in the absence of functional AGT, glyoxylate is converted to the insoluble metabolite, oxalate. Oxalate first accumulates in the kidney, leading to renal failure, and then leads to systemic oxalosis, depicted in the images on the right. Lumasiran is a GalNAc-conjugated RNAi therapeutic we're developing for PH1. It targets a gene called HAO1, which encodes for the hepatic protein glycolate oxidase, or GO. Knocking down GO prevents the conversion of glyoxylate to oxalate, allowing the soluble metabolite to be excreted. By blocking GO and lowering the levels of oxalate in PH1, we hypothesized that we could prevent further renal damage in patients who are early in their disease course and still have a residual kidney function. Moreover, we hypothesized that in patients with more advanced disease who have substantially impaired renal function, reductions in oxalate may prevent or halt the features of systemic oxalosis. But nevertheless, while there was a strong biologic rationale for the potential of lumasiran to benefit PH1 patients, there were a number of challenges we needed to overcome in order to successfully develop this medicine. First, PH1 is an ultra-rare disease with very few patients available for clinical trials. There was limited natural history data, and with no approved drugs, there was no regulatory precedent. Second, as I discussed, this is a fairly heterogeneous disease. The age range extends from infants to adults. Patients can have normal renal function all the way down to end-stage renal disease requiring hemodialysis. And there are multiple clinical disease manifestations. These, of course, really complicate the choice of endpoints, as there is no single endpoint that applies to all populations. Moreover, the rarity of the condition prohibits powering the study on clinical endpoints. The most obvious measures of efficacy are measures of oxalate, either in urine or plasma, given that elevated oxalate levels drive the clinical outcomes. There was no precedence for or acceptance of these biomarker endpoints to date. Furthermore, we had limited natural history data to define the minimally clinically important difference in oxalate levels, and the available assays were clinical diagnostic grade and lacked the sensitivity to be used as clinical trial endpoints. Fortunately, patient and expert organizations banded together to have points for the serious disease and create some consensus. The Kidney Health Initiative was a collaborative effort composed of patient, industry, and regulatory members that took on the task of points for trials of PH1. Early disease in patients with CKD grades 1 through 3A, they proposed a urinary oxalate surrogate. And in patients with more advanced, i.e. CKD stages 5, proposed plasma oxalate as a valid surrogate. Helping to define biomarker-based endpoints for this disease and for the full spectrum of disease was obviously invaluable and a huge contribution by the advocacy community that really path forward. In parallel, we worked to improve the assays. As I mentioned, the available assays were diagnostic grade. Because we were using oxalate as a surrogate endpoint, it was critical that we had a sensitive and reliable assay. We in fact developed an LC-MS-based assay for plasma and for urine that met regulatory requirements and would be able to sensitively detect changes brought about by lumasiran. We assembled a comprehensive development plan to address the broadest swath of patients with PH1, from adults and renal function to patients with and systemic oxalosis. The 3 pivotal trials are shown on the right. First was Illuminate A, and above. This was a random double-blind study versus placebo, 39 patients. Consistent with the KHI white paper, it was urinary oxalate lowering. CT was two single-arm studies. Illuminate B was in patients less than 6 with preserved renal function, to efficacy and safety in infants and young children at an endpoint of urinary oxalate lowering. It was Illuminate C. This was in patients of all ages who had advanced, as indicated by a GFR level 5, patients with ESR analysis. This is a single-arm study. Given these patients had significant renal disease, the primary endpoint was plasma, as suggested. All studies had primary endpoints with open label additional 4.5 years. This slide shows the result endpoint, change in urinary oxalate in Illuminate A, the placebo-controlled trial in PH1 patients 6 and older with preserved renal function. We observed a 53% reduction in urinary oxalate at 6 months in lumasiran-treated patients, and that placebo-treated patients had similar reductions when they rolled over onto active drug. As 30% lowering was clinically significant, this was a substantial response. Lumasiran was well tolerated in the Illuminate A study, with the majority of AEs being the most common AE, injection site reactions, which were mild. In Illuminate B, we tried to extend the results of Illuminate A in patients under 6. You can see consistent effects in patients across 3 body mass, all showing substantial reductions in urinary oxalate with lumasiran administration. Lumasiran was well tolerated in this population as well. In these very young children, the most again was mild and transient injection site reactions. I mentioned the involvement of the advocacy community to help define endpoints for this disease, but that was really just a sliver of what they contributed. We worked with a number of advocacy organizations in the US and in Europe. They gave us input on our development plan, helped get the word out about these studies, and advised us on what patients wanted out of a new therapy. A huge contribution was made by the Oxalosis and Hyperoxaluria Foundation in particular. They sponsored the first externally led patient-focused drug development, or patient voice, meeting with FDA. This was an amazing meeting where patients and caregivers were able to convey to FDA reviewers the impact of the disease on their lives, their hopes and needs for treatment, and their tolerance for risk. No doubt that this was incredibly helpful to the FDA, and particularly for such a rare disease that most will only have read about. Lumasiran was approved in the US and Europe at the end of 2020. And given the theme of this session, I thought it'd be helpful for you to see the press release that the FDA issued at the time of approval. Division Director Norm Stockbridge specifically cited the contributions of the patient advocacy community, physicians, experts, and sponsors, and the patient-focused drug development meeting as major contributors to the successful approval of lumasiran for PH1. As I mentioned, we have a third phase 3 study in patients with advanced PH1. This is a single-arm study in patients of all ages with GFRs less than 45, including patients on hemodialysis. We recently announced top-line data that the trial met its primary endpoint of plasma oxalate reduction, and that the most common AE was again injection site reactions. We'll be presenting these data at an upcoming scientific meeting. We'll be filing supplemental NDAs with these data to hopefully broaden the label to address this particular population that has a tremendous need for an effective treatment. So that concludes my presentation. Developing lumasiran as a treatment for this devastating disease, PH1, has been a tremendous source of pride for all of us at Alnylam and for me personally. And it's important to acknowledge that successful development of this medicine depended heavily on extensive input from and partnership with key stakeholders in the patient advocacy, physician, and regulatory communities, which led to a rapid and full approval based on novel biomarker-based endpoints. Thank you for your attention, and I'm happy to take any questions during the Q&A session. Enjoy the rest of the meeting.
Thank you, Pushkal. What a great example of rare disease success and collaboration. I'd like to open the floor for any questions from the audience, and I'll take a look through the chat and see what's come in. Okay, so perhaps the first question, there's a question about effects on kidney function. And so maybe more generally speaking, given that you had successfully negotiated the use of the surrogate marker, which showed really a dramatic response, do you have evidence yet of the ultimate evidence of oxalate lowering on end organ injury or function?
Yeah, no, that's a really important question and one we've been thinking a lot about. So we actually designed all three of those with long-term extensions. So they actually, while the primary endpoint was at 6 months, we're following these patients for 5 years. So we can document clinical benefits, hopefully, over time. We have actually started to see some very preliminary evidence of some benefits, some reductions in the first 2 studies I talked about, B, which is in patients with function, where we're seeing some reductions in nephrocalcinosis. So, calcification of the kidney basically has, you're seeing some evidence of trends in the right direction for calcinosis, as well as in the occurrence of kidney stones. It's small numbers, it's early days, but you're seeing this consistent pattern across both studies, across that full age spectrum. So that's encouraging. We're also going to look really closely at this population of patients with systemic oxalosis who've already, had substantial or who are in end-stage renal disease and now have systemic oxalosis. I think in those patients, we'll be able to hopefully see potentially some effects on end organ damage outside of the kidney, whether it's an ocular health, cardiac, bone as well. And so we're following all those parameters in those patients. And then there's a group of patients who may have some preserved renal dialysis, and we'll be able to hopefully over time see if their dialysis intensity is able to be reduced as come down in these patients. So, but some early signs that are really encouraging in terms of nephrocalcinosis and stones, and we'll follow them longer to see hopefully broader effects.
That's fantastic. And I just will comment that I think that that's a really important setting. I can think of many neurologic conditions for which this sort of approach is going to prove to be necessary, where there may be a surrogate marker, but a longer term monitoring for the ultimate clinical benefit. So that's very, very helpful to have as a template. Congratulations.
Thanks.
Another question from the group, from Rebecca Miles. What's the medical community doing to diagnose patients as early as possible? Is there newborn screening for these genetic conditions, and would that be beneficial to the program? And obviously, would this medicine be beneficial in that setting?
Yeah, that's a really important question. More than half of these children are diagnosed in infancy or childhood with this condition. And so it does occur quite early, and there are patients who are often found at the time in end-stage renal disease. It's extremely rare as an autosomal recessive disorder. There are discussions underway about whether this might be included in prenatal screening. The main focus that we have been doing is in terms of patient education and physician education. The advocacy groups have been really helpful here. It really is just like a lot of these orphan diseases. When there wasn't therapies available, I think there was a little bit of nihilism in terms of diagnosing this at times. And I think now is really increasing awareness that there are interventions that are and making sure that physicians know to look for this condition. It's actually quite easily diagnosed. We make genetic screening available, but there are also biochemistry can be done just looking for oxalate, for instance, urinary oxalate levels. So it's a lot of disease education to facilitate early diagnosis. And yes, maybe one day there will be prenatal screening possible as well.
That's fantastic to hear. It seems certainly like a situation where the tools exist, and now with the therapy in hand, that would be really exciting to push forward. Well, thank you again, Pushkal.
Thank you.
All right. For the scheduled full talk of this, I actually have myself. But before I actually give the last talk, and I'll spare you the self-introduction, I do want to point folks' attention to an on-demand talk, a very exciting on-demand by Haiyan Zhu, entitled Exon Skipping Antisense Oligonucleotide Therapy for a Patient with Niemann-Pick Type C. That talk, I encourage you to go visit the website and to view that talk on your time. I've seen it already. A nice complement to the themes that we today. So, to close out this scheduled talk session and to introduce us into the panel discussion, I have the pleasure of closing out with a presentation that I will share. And I'll go ahead and get that queued up here now. Okay. So the title of my talk is the N of One Collaborative. And I'd like to share with you in a talk that will parallel much of some evolving effort to help bring additional forces and resources on this problem of using platform ultra rare conditions. And as a scientist, I have to say that it's been one of remarkable privilege with the community, this community several years ago, 4 years ago, in fact, when I first met the patient Mila Makovich, who introduced me to the world of oligonucleotide therapeutics, introduced me to all of you, and led to a remarkable and really community-based effort to develop a drug for her. It was in 2019 that we published this report of a patient-customized oligonucleotide therapy for a rare genetic disease. And that effort involved many of the folks that you've heard speak today, and many of you who are listening today. And I'm incredibly grateful to all of your collaboration for that. In the years since then, we've seen continued further advances that really demonstrate that this idea is an idea whose time had come. We've seen, as you've heard in prior talks at OTS, US ALS, led by my colleague Neil Schneider, along with colleagues Bob Brown, John Watts, scientists at Ionis, and support from Project ALS. You heard today as well about efforts for C9orf72 ALS with an exciting antisense oligonucleotide advance at UMass by Bob Brown and John Watts. And there are many, many like this underway. And I'd like to, there's a quite diverse source of these efforts from academics like myself, like Bob and John and Neil. There are some really remarkable efforts put forth by foundations, and we're really grateful to the entrance of Lorem into the space and all that Stan is doing to advance resources to bring forward these types of efforts. And there, in addition, also led efforts too, where multiple parties, some novice, some more experienced, that are also stepping into the fray to see whether or not this type of technology might help their community. Just by way of example, one project that we publicly is our laboratory in the wake of our Mealison experience, where we designed and advanced an allele-specific ASO program for ataxia-telangiectasia. This slide shows a patient who has this devastating cerebellar neurodegenerative condition. And in a partnership with our lab and the AT Children's Project, we evaluated a series of patients who had various AT mutations and identified and designed an antisense oligonucleotide, and ultimately chose as the most promising an antisense oligonucleotide directed at a mutation that this young child, Ipek, carried. And working together collaboratively with the FDA, as well as other scientists in this community, we advanced this program through manufacturing, safety studies, and first patient dosing in January 2020. And that effort and the many others that I won't have time to mention really point to the topic of today's session: the use of antisense oligos as a platform for individualized or ultra-rare indications. And as we know, this leverages incredible foundational work done by visionaries in the field that have shown that ASOs are inexpensive to manufacture, remarkably simple to deliver in various tissues, and they have a growing and promising record and efficacy record through efforts of folks here. And of course, ultimately, their rapid modularity and their rapid customizability makes them especially suited for use in this manner. So why are we here? I've titled my talk The N of One Collaborative, and what is the purpose of that collaborative and that interest? And many of those driving forces have been mentioned in talks today. We are seeking to use these technologies to address unmet needs in ultra-rare disease out of a sense of social responsibility to try and address mutations that might be below, say, thresholds. We are also perhaps interested in the concept of individualized medicine, that bench to bedside drug development as almost a practice of medicine. That's an invigorating vision, and this seems still with closer than ever before. We are interested in the basic science, in scientifically efforts, basic ideas and hypotheses about mechanisms, about establishing proof of principle for therapeutic windows. Or perhaps we're also interested in this other challenge, which we all recognize in the genetic space, which is that with some 7,000 individual genetic diseases, how on earth will we possibly manage for any of them? And perhaps a rapidly customizable approach might be an alternative way of thinking about this problem, if we can categorize these conditions as amenable to various mechanistic, those that might be amenable to splice rescue, to knock, or perhaps through other technologies, nonsense read-through or genome editing. Another reason, though, why we're here is that we know it's not going to be as simple as all of that. We know that this technology has been described appropriately by experts in the field as possibly deceptively simple. And we know that despite the fact that drugs in this category are more similar to one another than any two small molecules, any other family of therapeutic modality that has preceded it, there are still sequence-specific effects that have to be accommodated. And that means that there are many hazards to navigate, scientific, clinical, but also regulatory, logistical, financial, legal, ethical, patient safety and expectations. These are all paramount. And there's a lot of work to be done, and it's important. Slide I showed a couple of times at OTS, but it's a recurring mantra in our group. The idea that this is no cowboys are no John Waynes, please. Now, back in 2019, I was actually on a train heading out to New York with a dear colleague and friend, the mother of Mila Makovich. And at that point, we were talking about what's necessary to make this field move. And I slide, which shows in concept a collaborative that could allow various parties, such as Academic Medical Center, which began the Mulison work, other parties, other academic medical centers that might also advance this type of work, and other parties too, perhaps businesses or foundations. And that a hub would be helpful to help facilitate communication between these groups as we try this incredibly challenging thing of deploying this platform technology in a way that benefits individuals and those with ultra-rare conditions. And from that hub, that concept, we have built out this concept of furthering a model for collaboration that was really based off of how the community gathered around the Mulison effort. The analogy that I'd like to throw out to you is one perhaps fanciful. Barn raising is an action of a community, which a barn for one of the or rebuilt collectively by members of the community. And dating back to my days as an 18th century rural North American farmer, a barn was, but it's also a large and costly structure, the assembly of which required more labor than the family could provide. And to address this need, a barn raising enlisted members of the community, unpaid, to assist in the building of their neighbors' barns. And because each member could ask each other for help, reciprocation could eventually reasonably be presumed for each participant if the need were to arise. And it's in that spirit I am discussing this topic of a new collaborative with all of you. For history's sake, back in May 2019, we had a meeting with the FDA, a critical path innovation meeting, to discuss this topic of how this field, once the door had been opened through Mielison and similar efforts. And at that point, we proposed a pilot in which this type of effort needed to be coordinated and the experiences aggregated to demonstrate repeatability as well as to work. And that mission also was furthered by the OTS with Art, Krieg, Anamika, and John, along with others, the OTS N of 1+ force. And actually, this group, which I was proud to be a part of, also had planned a workshop to actually discuss these issues and to make traction back in April of 2020. Of course, we all know what happened in the spring of 2020, and plans for that were waylaid. In the meantime, the entry efforts like and Stan's incredible generosity, coupled with the scientific team and donated time of Frank Bennett and other scientists at Ionis, injected lots of new hope into this space. And Lorem, in partnership with that are ready to receive the drugs that it's making free of charge, medical centers like UC San Diego, Rady Children's, Children's Hospital Colorado, and our hospital, and many others that stand to benefit from this, are working towards making this all happen. At the same time, there are also efforts and I'll highlight involves our own current president, Anamika Arts-Marus, who of the Dutch Center for RNA Therapeutics, an effort to the first European center committed to the development of these medications in Europe. And there are similar efforts in Asia and other countries as well. So in all of this context, the releasing initial guidances with a procedural draft guidance, toxicology draft guidances released in April, and further guidances to come, we held a workshop of what we call the N of 1 collaborative in June of this year, with its mission to make individualized genetic medicines accessible to patients worldwide. The values here that we are trying to espouse, our drive center values, are expertise, but also bringing in an inclusive approach towards welcoming the community and to establish by which these best practices can be refined, criticized, revised, and reviewed. And our focus here is on treating patients and sharing the learnings from treating patients. In that workshop, we had some 50 participants in June, representing 32 institutions from around the world. And there's an asterisk here to point out for our is that there's much more to just ASOs that has to be done through a group like this. The type of expertise and standards and approaches that need to be formulated for the delivery of medicines, the selection of patients, and the care of patients receiving these individualized therapies is a challenging problem that will apply to many other potential platforms that might include or CRISPR genome editing or mRNA therapies. So what can we do together? We can collaborate. We can establish community expectations and standards. We can ensure clinical trial readiness, prepare our institutions to actually deliver the care that, that is going to be created by the design and manufacturing. And that this type of work is necessary to address both opportunities and challenges that may actually go beyond just making the medicine. What else can we do together? We can work with stakeholders to nurture this field, of course. We can pick good cases. we can avoid avoidable mishaps. Jesse Gelsinger is a famous patient whose early demise led to a major setback in the advancement of viral-based gene therapy. We can build patient registries to minimize duplication of effort. We can share data and learnings through case conferences and clinicians and investigators sharing their experiences with the treatment of these patients to make the dissemination of knowledge faster, to build safety databases together with parties like Lorem, to cross-reference one of these efforts can build upon each other necessarily. The collaborative is dividing itself into a number of work groups to tackle some of the issues that we've identified. First, a work group based around patient selection, i.e. who gets these drugs or what conditions are serious enough where the risks and benefits might merit such an approach. A very practical institutional implementation work group that helps investigators prepare their institutions for all that's necessary in the drug itself. The issues of with what consent these efforts need to be put together, with what oversight required to make sure that this is done. Research and clinical agendas are not during these efforts. Outcome measures, we can assemble clinical expertise, learning from lessons, perhaps like the Angelman's community, to ask whether it works, and then also to be able to aggregate clinical and preclinical safety data to identify and inform each other about what hazards there are to watch out for. Finally, questions of ethics and equity. How do we make all of this accessible and sustainable? This topic was highlighted in Stan's talk about Lorem as well, and we look forward to working together with them to tackle these really challenging questions. This workshop series is fall 2021, and with a number of work in the community as shown on the slide below, Mathis, Rick, and Jonathan, among the many people who are contributing. And just by way of example, we are drafting a patient selection publication that will help make transparent the criteria with which patients with various genetic conditions can be triaged as to whether or not this approach might possibly work for them. How to get involved? I encourage anyone who's interested in this new and challenging space to contact one of the members of our current de facto steering committee. These are all volunteers, but Anamika Artsmarus, Keith Flanagan, Art Krieg, Mathis Sinopsic, John Watts, myself, and many are among the many folks who are having these conversations and helping get this nascent effort up and running. I also encourage you to email the organizers of this conference, in particular Jerry, to join our email list so you can be informed about workshops as they come up. With that, I'll thank you for thank the many folks involved in this evolving ecosystem that are invested and are thought partners in all of this work. I'd like to acknowledge the support of the Baker Family Foundation, which has seeded a lot of our initial efforts as contributions from all parties involved. Thank you so much for your attention, and I will close right there. Okay, and I'm going to waive any question and answer sessions following my talk, so we into the panel. And it's my pleasure to introduce our last to the panel. Our last speaker is Marika Kimbrell, who's joining us from DC. And Marika, director at policy in drug evaluation and research at the federal at the FDA. Prior to that, served as deputy chief of staff to the FDA commissioner. So Marika, thanks so much for joining us.
Thanks for including me.
I'm going to actually start with a question to all the panelists, and I'm going to start with you, but it's a very simple question. I'd like to have each of you respond from your perspectives. What do you think is necessary to make that you've heard described today a success?
I'd say, Tim, that your presentation, I think, hit the nail on the head. I think it's collaboration and coordination across a large group of people, and I think we've seen a lot of that in action in the presentations today.
Thank you, Marika. I'll come back to you with more questions about upcoming guidances in a little bit, but with that for the moment then. Thank you. Let's turn that next question on to Stan. From your unique perspective, what do you think is going to be necessary to make this all work?
Well, I think, a primary therapy to treat the patients you can today while investing in creating treatments that are better and useful. And so the question is to focus short term the problem, concerted entire community toward a more holistic. We need to begin solving the diagnosis problem, and I believe strongly it's in the interest of all the to encourage adding genetic characterization to newborn screening with appropriate. Certainly don't want to repeat the eugenics experience, and then models in the roadmap that's being created and bring other organizations and other technologies on. This can only succeed if at some point we have a system-wide healthcare system that we all and so that is my major goal long term, is to play a role in driving these that will be holistic. The number of patients we're losing every day simply because they can't find a way into being referred to the proper center. I get emails 5 times, and it's maddening. We have to fix this.
Thank you, Stan. I agree with you. Finding ways to solve that bottleneck at the front end to find these patients quickly and in time is so critical. Thank you. Then Bob, what would you say?
Yeah, so it's hard to improve on the comments. I mean, I think there are a few critical points here. One is being very certain that we have what you might call precision biology. We have many instances, for example, in ALS, where there are some genes that are said to be both acting by loss and gain of function mechanisms. And I think we need to be very precise about how are we really seeing, when we have only one mutation, in fact, the true genotype, or are there intronic mutations, for example, the physiology. At a much more sort of nuts and bolts level, I think ease of manufacturing is going to be an issue, GMP manufacturing. I think finally, in terms of the cost structure, which we as academics have to struggle with, can we find a simplified way to come to agreements about animal testing? Must we do, for example, GMP rat testing, or are there other ways diminish the financial burden? I think those are three of the areas that we struggle with as academics, at least.
Thanks, Bob. Certainly, especially that last one, that we all know that the costs of animal GLP studies are tremendous. Efforts by Lorem notwithstanding, it does remain the current single most rate limiting step. Let's Liz.
So I really agree with everything Stan and Bob said. Those are critical issues. But even at a more basic element of seeing a patient in clinic, and like you have to jump through many hurdles times to even get exome sequencing or find out what the patient's genetic problem is. As Stan said, having some opportunity to be able to screen patients, that would be great. But even patients who have symptoms and trying to identify their disease can be very challenging because of insurance restrictions and problems with getting approvals for testing. And so I think we need to be able to basically test people, and also recognition of symptoms. When a patient, we talked a little bit about finding patients early in disease, and really that's a critical thing. The neurological diseases that we face, if you treat the patient when they're, floridly with anything, it's not really going to work that well. You have to get these patients, to get them when they're first showing signs of disease for these new genetic and other, molecularly based strategies. And so we need of not just saying, this just may be normal variation. Let's watch the patient longer, because that's what our culture is right now. I think we really need to change that if we are going to have strategies that are really going to reverse diseases and make and have that huge impact that we want to have.
Thank you, Liz. I might say I couldn't agree more. I on a recent ACMG practice committee that put forth the first recommendation that exome sequencing needs to be done for intellectual disability and congenital anomalies. And that's frankly something that, in my mind, we've known for 10 years. But here we are in 2021 now, recommendation, and hoping that insurance companies. So it does seem like a critical rate. Thank you for that, Liz.
I want to add just one more. Degenerative diseases, virtually in children, virtually always developmental delay and look like they're going toward an intellectual disability. And having that genetic testing available is an opportunity for us to intervene early in those diseases before the patients are actually degenerating. So I think that's a really important strategy for those.
Excellent point. Excellent point. Pushkal, what lessons did for us from might some help coming from SI?
Sure. I mean, I guess a couple of things. I think points made by everyone, and I guess I want to start by complimenting Stan and Tim and all of you, and Lorem. I mean, you guys are the first barn raisers, as you said. That's how we're going to move this forward. So compliments to all of those efforts. I really applaud them. I think what needs to be built. I agree with all the points that have been made. I think what's really great is there needs to be this basic level of infrastructure fostering in terms of preclinical work, et cetera. Some standards, I think, in terms of how this is. I don't think anyone wants these N of 1 all come across. That's not the intent here. It's really to make sure that there's some understood methodology behind which diseases are being selected, what preclinical work and et cetera needs to be done, how are assessments being done, how's benefit risk being assessed. And I think that infrastructure and collaboratively, I think important. And as you said, it takes all stakeholders in fostering that. So I think that's the most critical element from my perspective. I think siRNAs, siRNAs offer also a very modular oligo-based platform, and so absolutely, I think there is I think from a technology perspective, right now, we're so the technology primarily to really silencing. It doesn't sort of more allele specific at this point, although those technologies, those opportunities may come forward. So I think over time, though, I think as the applicability of siRNAs broadens as well across some of these other aspects, of modulation, I think that certainly I would imagine siRNAs would be another tool in the armamentarium initiatives.
Thank you. The idea is this knockdown absolutely no. Perhaps Dan, I'll invite you to comment if you might. Attractiveness of that in the case experience within Lorem. Sitting on the Access to Treatment Committee, I know that the knockdown seems to be a critical part of what we're trying to do.
If you don't mind, Tim, I'll back up just a little bit and give you a more general specific. NLRM exists. I think these patients need help and I can help them. And we can't do it alone. And I think we've been remarkably successful in putting a very broad network of collaborators and donors. And we've just begun. And so there's a great opportunity to create human beings who have technology at their disposal. And so as new technologies mature and are ready for use, they should be brought on board. You know, as soon as I found it in Lorem, I picked up the phone and called every CEO to me. And you know, as gene therapy moves along, that's a critical element that needs to be added here, because neither siRNAs or ASOs can solve null mutations, and null mutations are a frequent occurrence. So we need to expand our network, and we need to do it efficiently. Duplication of effort, as you say, is a waste. Every dollar matters. And if you look at what we've done, we've reduced the cost per patient by 40% by just talking to CROs and so on. So take advantage of what we've done. You don't have to, but work alongside of us and let us help you. And so now specifically, what are the technologies? Well, obviously is the major missing piece here. And I think the people I talk to generally agree with me that it's not quite ready for broad application, but there are specific IH is trying. So we need to move gene therapy forward. And obviously siRNAs and other oligonucleotide approaches are very, and so as they mature, they should move into this work to do. And on occasion, there are targets that, you know, we can design single-strand ASOs to take advantage of AGO2, and we do. And we can also use siRNAs. And so if we run into a target that, you know, would work better than H1 to knock down, well, we'll do it. And so I would welcome Alnylam. And I would say, you're advancing your technologies, how about helping me? How about helping in Lorem? We need all the help we can give, including donors. Many applications already. I thought we'd have 4 or 5. And so we're expanding rapidly, but we need all the help we can get. So should come, other antisense approaches should come. Gene therapy must come. Small molecule repurposing generally is inefficient and doesn't really lead where, but if there's a drug that's has logic for it, it should be added to it. With us or with us. Either way, it doesn't matter. Just get involved and help us. Help us help these patients. So that's a long-winded answer saying I agree that gene therapy and siRNA need to continue to. I think quality is critical. And listening to this, I think people don't understand how rapidly antisense is evolving. And, you know, we're moving to new designs that get rid of toxicity. We know how to do it now. Also have information on innate immunity and how to reduce and ablate innate immunity. And all those things into ASOs as we speak. Talk to us. Let us help. That's my
Fantastic. Thank you so much, Stan. If I may write on to a point that you raised there, in experiences being and then also forming this nonprofit, do you have any advice for other also, as you say, want to also their charitable efforts to help this space? Are there any challenges or conflicts or concerns that you can reassure folks about who might want to do similar efforts?
Well, extremely charitable for a reason. We don't file patents. We don't do, I need to be clearly separated from my own. And so to those who are interested, still the technology you have needs to be matured before you're ready to take these patients on. And I tend to agree with that. Then help us while you're developing, and then take advantage of what we've invented if it seems to work, because the real issue is quality risk benefit decisions. As you know, participate in the AT, know how tough those decisions are. And then quality ASO or quality siRNA or quality gene therapy or small molecules. So experts on board, but don't try to recreate the knowledge they have. You know, a half-life in the central nervous system when you're giving intrathecal, you want 3 months, 6 months, and we can do it. And, you know, yes, you can put phosphodiester's in, but if you'd asked, I'd have told you put a methoxy at position 2 in the gap and you'll fix it, but okay. Don't recreate expertise that already exists, use it. And there are no barriers, there are none. And Lorem is entirely separate and a pristinely non-profit organization.
Thank you, Stan. All right, we'll take a couple of questions. Does anyone help add to that topic? If not, maybe I'll turn to some questions from the audience here. Question that is from Anamika. Choosing the right outcome measures is critical for these efforts, right? And having something that has some chance of demonstrating an objective reality is so critical. Can folks elaborate on how in your experiences, patients were involved in selecting or developing the that you've relied upon in your work, especially as some of those tools are really just being developed. And maybe I'll point this one to, because I know that is one of the key contributions of the Angelman effort.
Yeah, I think in Angelman syndrome, there was a strong effort to pull families in and to interview them and to identify what are the key problems in Angelman syndrome so that one could focus on those problems when moving forward. And then those were incorporated in global impression scales, as well as the ORCA is a measure designed to look at communication in impairments of communication that no language scale would ever be appropriate and would take a long time to move. And yet it incorporates key elements of communication, said, would quality of life. So I think that was a very important effort. We have other kinds of efforts that are very in Fragile X syndrome and Phelan-McDermott syndrome and Rett syndrome, where the community is providing feedback about what's most important to them, kind of making designer look at that. But this is where you have a number of patients. If you have an N of 1 situation, it's more challenging. And I think that really is a individual, and you'd love to have something objective you could really feed that would really identify whether the disease was improving, but oftentimes you don't have that diseases. And so it's really valuable information on the trajectory of what the patient did beforehand, and then be able to look at doing N of 1 trials. That is the critical analysis, is what was the trajectory before the patient started treatment and what is the trajectory after. And even for rare disease patients, a humongously very valuable thing. Although I think a lot of these diseases have such that you might not choose the same thing for each patient, and that's been, really a struggle for some of these diseases. So, well, picture of what will assess the disease best.
Liz, I mean, I think your experience is so valuable here. It's at once inspiring and also a little. I know how much work went into the development of ORCA and those women's community. Speak to the fact that to be full, that there's a lot of work to be done, trial readiness along the way that the community will have to gather around. But if I may quote, we do what we can right now, and we help where we can. Go ahead, please, Stan.
There is a literature on trials. It's there, and they're all crossover design, and crossover designs won't work here. And so at Lorem, a solution I think is working. What we do is we work with the treating physician or parent to define primary, secondary endpoints. We focus strictly on clinically validated measures because biomarkers in a single patient, you're never going to validate. And then during the 12 to 15 months it takes to get the ASO ready, we ask the investigator and parent to perform a natural history study with measurements that are clinical practice. Then when we're ready to treat, we have those measures, and we at least have a 1-year natural history that immediately precedes treatment that we can compare. That is a solution. I think it works, and it works in a very difficult setting where most of these are ill and are very long duration. So that situation where you have a desperate patient who's progressing rapidly, who's already advanced, and very crossover designs work, but the solution we have, I think, is working, and you're participating in that. And so again, we've thought through it, right? And we have a solution, and if someone can come up with a better solution, that'd be great. But
Stan, thank you. I concur with that. That is exactly the right framework. I might say it does sort of engaging the clinician and the patient. Of course, then there's the matter of what the clinician comes up with and what do they actually negotiate, which is not always easy, but that is the right way. I completely agree.
The big challenge is developmental delays, right? All these things space have developmental overlays. And if you think about all the things that we're going to be learning, among them, how plastic is the
Exactly.
and the challenge of understanding what the primary mutation is and what are modifiers in those two twins, the FUS twins that we helped Neil treat. Same mutation, big background, as far as anyone can tell, they're identical. Disease was 7 or 8 years later in one of them versus the other. How? Well, I mean, now we have an opportunity in the clinic to understand some of those. Just pay attention and do our work right.
Absolutely. Thanks, Stan.
I think this is absolutely the right approach. And this is the approach that we've tried to take with a few of our expanded access protocols for things that are not ASOs. I think that pre and post look at what the patient is doing, individualized basis is I can't think of any other way to do it in these patients.
And the key thing is to treat what's important. To know why you're treating, what you're treating, how you're going to measure it, and do it the right way. Anything else is doomed to failure, potentially do harm.
I love, agree with everything that's being stated. I'd love to actually pivot a bit to Bob and ask, Bob, you have a unique experience with adults. Many of the efforts that we're talking about are with kids who to communicate to us what's valuable to their quality of life, to their disease course. And we're working with parents as proxies. True for the cases that you're forward with. And so you have the opportunity, maybe are there lessons to be learned from conversations with your adult patients about how to craft these measures into meaningful outcomes that impact their real life function?
Yeah, the lessons from the adult population are what Stan has articulated so well. I mean, in each of the cases we've been involved with, we've wound looking at pre-disease course as the primary clinical measure of outcome. Clearly, it's best if we have something that's quantifiable, such as, for example, the poly-GP levels in CSF. But I think the overall is the same. I do think what the adult patients provide, of course, is a sense of quality of life and which sorts of indices or measures of quality of life one can the mix from the patient's perspective. And as you know, that is in virtually every study we do, certainly across the board. But I don't think the general principles are actually that different from what Stan has articulated.
Thank you, Bob.
And
Let me
by the
move
way,
to-
the central nervous system, the organ.
Yes, there are other
And that's why
opportunities.
we're, and these things happen in all organs. And so we need to focus beyond the central nervous system as well.
I'll turn to a question now from one of the audiences, and I'll question. So here's a question from Mina. Executing a comprehensive tox strategy is very expensive for patients. And this question will go to Marika, and I think then appropriately to Stan. First, what's the regulatory perspective on on-target pharmacology, safety pharmacology studies, and reducing toxicity studies if the molecule is from the same class as and has been approved for other indications? So that's a question about class-based approvals and then combining it with the previous question about sharing experiences from safety experiences with a certain class of molecules. Previously asked, are there safety all of the information that will be generated through Lorem, and will those be made available to everybody or only within the foundation's use? So Mark first and then Stan.
Sure. Yeah, I can tackle the first, but we'll leave the second for Stan. We're full aware that the pressures on tox data, which are critical, are difficult also for folks in these circumstances to produce. So some of you may be aware, but earlier this year we issued a guidance on this topic, trying to provide some more clarity for sponsors in this space, and have put forward a flexible approach to help sort of speed development along here where appropriate. And a lot of this in the details, while I think the guidance is helpful, involves ongoing discussions with FDA. And so we do encourage sponsors when early in development to come and talk to us and find out what will work in a given circumstance.
Thank you, Marika. Stan, do you want to tackle the second one?
Yeah, well, first of all, I would encourage everybody to read the guidance, and I thank the FDA. I think the response from the FDA has been extraordinarily rapid and highly constructive, and I look forward to additional guidances coming from them. But I think the limited tox studies that are required are about right, and all the CROs, and they're all doing these studies for us for cost. So, I mean, people are working on this. On the, again, you have to be sure that you don't do any harm. there are real issues here. There are risks, and we have to be prudent. And Tim, I forget the question you wanted me to answer. Sorry.
Well, I think it's a good one and potentially a challenging one. About the being able to share openly the experiences. Are there any limits to being able to share openly these knowledge and the knowledge that's generated?
I don't think there are limits. I think that's silly. First of all, at Ionis, we published, and the FDA has access to them, safety databases that integrate all safety observations from non-human primate through all controlled trials for every chemical class we're developing. We published it, and continue to do that. In fact, I think I'm the first author on all those papers. And at we are planning, and first of all, we encourage our investigators to publish cases. Very important to get those out, as you've done. We will publish annually an aggregate experience. We plan annual meetings of investigators and parents. And we are developing. There's one of the problems is there's no relevant advocacy group for. And so we're going to try to group around Lorem, around for these patients. Really, experienced is that there are wonderful people involved doing incredible things, and some training at all. It's really quite remarkable. And so later this year, I'll begin an Lorem podcast series. And among the things I'll try to people think about drug and drug development. There's no limit. Anybody who thinks is
if I may, if I may, and I pardon to cut in just because time is short. I think probably there's a question about whether preclinical findings that were generated in developing the drug. Well, I think that that's something academics feel quite strongly that they can share. But I would understand if it perhaps might feel out of to some other parties.
No, I mean, why would it be out of bounds? I mean,
that's wonderful to hear.
My problem with doing it would be, does the person have enough knowledge to interpret the data? Perfect.
Okay. Thanks. You know, I'm getting signals from the organizers that the next session has been postponed a little bit to give us some fantastic discussion. So I will probably last comment from the field, which is I do want to highlight that a note from Takanori Yokota from Japan, who that is really valuable to discuss and share this various approaches to this in various countries, including through the Japanese Therapeutics Society as well. And I could not agree more that what we are doing here and beginning here, different implementation strategies all across different models. And so I think that that's a really critical point. Thank you for raising that. With that, perhaps a final thought, I want to just thank all of our panelists and our speakers for a really illuminating session. I really enjoyed it's just amazing to see. And thank you all for contributing the remarkable. If anyone would like to, I will be joining the spatial chat, which is a chance in the break before the next session to continue conversations that we couldn't get to, and invite my speakers and panelists.